Good morning, everybody. Your presenter of this tutorial is Mr. Midamo TK, a biology examiner. Today we are looking at KCSE Revision 1997 Paper 2, Biological Sciences. So, welcome back to YouTube channel, Smart Traveler Memos. Welcome to our KCSE Biology Practical Revision. Today, we are delving into a hands-on biology practical. In question 1, where we have specimen R, a ripe banana, and specimen S, an intact carrot tuber, we will explore their plant parts and find out what nutrients they contain. In question 2, we will explore a real flower dissection using a fresh picked banana inflorescence. We will identify floral parts, determine its pollination agent, and draw a longitudinal section of the female parts. And finally, in question 3, we will examine two important mammalian organs, the trachea and the ileum. We will name the organ and the system of the mammalian body from where each specimen was obtained. We will state two observable similarities and one difference between specimens G and H. We will also state how each specimen is adapted to its function. But before we go to the questions, so before we look at the questions, let's look at the confidentials. So in question 1, you needed a ripe banana fruit labeled specimen R, an intact carrot tuber labeled specimen S, iodine solution, Benedict solution, scalpel, 4 test tubes, a test tube block, a white tile, Bunsen burner or means of heating, a glass rod, and a dropper. In question 2, the confidential asked for a freshly picked flower obtained from banana inflorescence. Note, a banana inflorescence is the heart-shaped structure in a banana plant which appears when bananas are to be produced or when bananas are developed. An inflorescence has numerous flowers. A piece of mammalian trachea, about 4 centimeters long, labeled specimen G, will be required for question 3. And again, question 3 will require a piece of mammalian small intestine or ileum, about 4 centimeters long, labeled H. So, that is specimen R and specimen S for question 1. All right. You can see the ripe banana fruit and the intact carrot. You can see them, the ripe banana. And so let's come to the questions. Question 1. You are provided with specimens labeled R and S. Examine them. Which part of the plant is specimen R? Give a reason. S, give a reason. Make a transverse section of specimen R. Draw and label the sections. 1C. Using the reagents provided, carry out food tests to determine the food substances present in specimen R and S. Record the food substances tested for procedure, observations, and conclusions in the table below. So that is the table. Specimen, food substance, procedure, observations, and conclusions. Specimen T for question 2. A freshly picked flower obtained from banana inflorescence. Question 2. You are provided with a specimen labeled T. Examine the specimen. Describe the floral parts of the specimen. From your observation, suggest the possible agent of pollination. Make a longitudinal section of the female part of the flower. Draw and label the section. So I just found that the specimens had been interchanged. All right. So we have G, a piece of mammalian trachea, and specimen H, a piece of mammalian small intestines. And the questions are, you are provided with the specimens labeled G and H. Examine them. Name the organ and system of the mammalian body from where each of the specimens was obtained. Specimen G, system, specimen H, system. B. State 2 observable similarities and 1 observable difference between specimen G and H. Similarities, difference. Using observable features, state how each specimen is adapted to its function. Specimen G, specimen H. Now, we are through with the questions. The next thing is now to look at how we as examiners marked this question. Remember, guys, I participated in this marking. So, which part of the plant is specimen R? Is a fruit. And the reason is that it has 2 scars, and the scars are scar for attachment on flower stalk or pedicel, and scar left by flower parts, that is the style, petals, and corolla. S is a root. You can also call it a root tuber, or main root, or taproot. And the reason for this is that the scar for lateral side is seen. There are secondary roots which can be seen, presence of lateral roots. Then we have the scar of stems. And then we have main root with the lateral side or lateral roots or secondary roots. Making a transverse section of specimen R, draw and label the section. And there you are. we have the transverse section, and the rebels are mesocarp, epidermis, placenta, endocarp. So you can have any of those two diagrams around. You get 3 rebels. And because you are making a section, I don't think there was drawing mark. Recording the food substances tested for procedure, observations, and conclusion in the table below is where we are now. Let's try to move this a bit. Yes, yeah. So, we have specimen R, part 1. The food substance is reduced in sugar. You add a few drops of Benedict solution and heat it to boil as the procedure. It turns from blue to green, yellow, then orange, and therefore you conclude that reducing sugar is present. The second food substance tested was starch, where you add a few drops of iodine solution, turns blue-black, starch present. For S, reducing sugar, the same procedure, where you are adding few drops of Benedict solution and heat it to boil. It turns from blue, green, yellow, and finally orange, meaning reducing sugar was present. For the starch, you add a few drops of iodine solution and it turns blue-black, starch was present. We are through with question 1. Guys, welcome to question 2. In question 2, where you had been given the flower, describe the floral parts of the specimen. Petals are 6 or 5, some fused, or these are points. Yellow or currant has 1 modified keel or rounding stage. Stamens, notice that the semicolons are the marks, are 3, they are infused, there are 6 or 5 stamen, and a developed vestigial stamen. Anthers are long or orange, have the length of the stamens, short filaments, have nectar guides, stroke, half grooves. All those ones, you give 1 mark. Yellow colored anthers or filaments, you get another mark. Pistil, the ovary inside, or the ovary is inside receptacle. We call that one hypogynous ovary. Inferior flower parts arise from above the ovary. Stigma is lobed or crab-shaped and sticky. Yellow colored, same level with anthers, style long. All those are marks. You get a maximum of 13 marks. No. Okay. Sorry. And going back. So, question 2b, from your observations, suggest the possible agent of pollination, and this one is either insects, birds, or bees. All right. C, a longitudinal section of the female flower part of the flower is there, where you have the following rebels, stigma, style, ovary, and receptacle. Question 3, marking. Name the organ and the system of the mammalian body from where each of the specimens was obtained. Specimen G is obtained from trachea or windpipe. System is respiratory. Specimen H is obtained from ileum or small intestine or duodenum. The system is digestive system. State 2 observable similarities and 1 observable difference between specimen G and H. Similarities: presence of muscles, hollow, have rumen or tubular. Difference: cartilage rings in G, no cartilage rings in H. G is open and H is closed. Continue with C, part question 3. Using observable features only, state how each specimen is adapted to its function. Specimen G: presence of cartilage that keeps it open, prevent it from collapsing. Guys, the other time I told you when we put a stroke, it means an alternative. Presence of muscles between cartilage to allow movement or elasticity. Hollow, have lumen. Tubular, to allow passage of air. There is a spelling mistake of tubular. So I was correcting the one to tubular. So that's where I did it. Hollow, have lumen. Tubular, to allow passage of air. Mucus, to trap dust particles, germs, particles in the air. H, hollow or tubular lumen. Hollow or tubular or lumen to allow passage of food. Presence of muscles to allow peristalsis or elasticity. Inner walls folded to increase surface area for absorption. So we were also making a correction there. Inner walls folded to increase surface area for absorption. Presence of mucus to reduce friction, protect inner walls against digestion. Superior. I conclude by telling you this. Thank you for joining this practical session. And remember, the best way to learn science is to do it hands-on. And that's it. You have just completed a full flower analysis of a banana specimen, which is perfect practice for KCSE Biology Paper 3. Understanding the structure and function of the mammalian organs also helps you master practical biology. Keep revising, and all the best in your KCSE exams. Please comment below with questions. Like, share, subscribe, and hit the bell for more KCSE biology guides. If you find this helpful, give a thumbs up, subscribe for more practical tutorials, and don't forget to download your free revision sheet in description. See you in the next lesson. I extend my deepest appreciation to the Kenya Examinations Council for providing original examination questions as the basis for this work. I also acknowledge the valuable contribution of ChatGPT, whose assistance in refining and enhancing the clarity of the presentation was instrumental. I'm sincerely grateful to my colleague examiners for their professional support and collaboration throughout this process. Finally, I wish to express profound gratitude to my family for their patience, understanding, and steadfast support during the many hours devoted to the preparation and the typing of this material.